My project worked on investigating the role of Cyto2 and PC3 cell migration and P53 expression using siRNA knockdown. Prostate cancer is one of the most diagnosed cancers among males, and its treatment success can vary depending on whether it metastasizes or not. siRNA are small molecules of RNA that are able to knock down gene expression and alter the expression of genes. P53 is considered the master regulator of the cell cycle and has been linked to many cancers, including prostate cancers. It was hypothesized that knocking down Cyto2 would reduce the expression of P53 and also reduce cell migration in PC3 cells. To do this, PC3 cells were cultured. They were also treated with either siRNA against Cyto2 or siRNA that was a vehicle control. That was a negative control. So the cells in replicates of 4 were, for PCR, were treated and with either of these two and analyzed using technical replicates for both genes. Also, I did a test to look at comparing the times just to see using the negative control because I had a fluorescent tag to see if transfection had occurred because there's this transient transfection. Also performed a Boyden chamber assay to test cell migration. So the results. So while I was not able to space not show a picture, this image showed that at 72 hours, it looked like not really much transfection, and based on using image data to count the cells, I found that basically almost a similar percentage of 40 and 43. So because I didn't have any end numbers, I said that's not a lot. So that looks like transfection did not occur. So looking at the Boyden chamber, which I started at 48 and 72 hours, showed that there was not a significant difference in the migration between cells that were treated with the siRNA vehicle control or the cells treated with the actual siRNA that knocked down Cyto2. While there was less overall, the difference was not significant, its p-value was 0.9. So, within the timeline I used, that showed no knockdown. The PCR result also conclusively showed that at 72 hours, Cyto2 was not knocked down, as the values were exactly the same, including having a p-value of almost exact same, 0.4. And so P53 expression was basic, not significantly different with 0.2, and that makes sense because Cyto2 was knocked down. So the results indicate that the timeline I picked, it seems like Cyto2 was not knocked down. In the PCR, that was definitely not the case. However, the Boyden chamber, you wonder, could that be the time because it's transient expression? Maybe during some of that time you started seeing it some, which is why cells are able to migrate, but not as many because couldn't the whole time. So that's why there's a difference, but it's not significant. And so that's why I can't say anything about P53 because if the cells were not, because while there is no difference, there's no difference in Cyto2. So I haven't shown that knocking down Cyto2 has no effect because I never knocked down Cyto2. And so this would be for looking at future work, would be I'd actually want to try and alter different times because I did find while trying to come up with an idea, an article that said they had success looking at 24 hours for knocking down Cyto2 in prostate cancer. So if given the opportunity, I want to try and repeat these results with the PCR using 24 hours in the Boyden chamber. Maybe you could try 0 to 24 hours or 24 to 48 hours. And look at the results to see if knocking down Cyto2 would affect migration and if it would affect P53 expression, because right now at one time point, because I've never knocked it down, I can't definitively answer to say that. And I would like to thank the help of Jessica Livingston, Michael Gibson, Chris McCauley, Marla McKenzie, and Alea Quilty for their help and support throughout this process.